Dear students, this is Dr. Manikandan, Assistant Professor, Department of Life Science, working in Kristu Jayanti College, Bangalore. So the topic of today's discussion is B cell and the T cell. So where this B cell and T cell originates from? They originates from the hematopoietic stem cells. The stem cells are the mother cells from which every type of cells arises. So the process of the proliferation, differentiation and maturation of any kind of blood cell is called hematopoiesis. So this hematopoietic stem cells they reside in the bone marrow and they are being regulated by several factors which include cytokine and the growth factors. These hematopoietic stem cells are pluripotent, that is, they have an ability to differentiate into any kind of cells. If you see, the report says that the human body blood stem cells produces approximately 10 billion new white blood cells every day, and that we call it as an immune cells. So these hematopoietic stem cells are self-renewing, and once they undergo a differentiation and maturation, and thereby producing a specific type of blood cells. Okay, so this hematopoietic stem cells can be differentiated, okay, into a lymphoid progenitor cell or myeloid stem cell. If you see the immune system, cells of the immune system, it can be divided into two group. One is the lymphoid cells, the other is the myeloid cells. Lymphoid cells can be further divided into a T cells, B cells, and natural killer cells. And myeloid cells can be differentiated into granulocytic cells, monocytic cells. Granulocytic cells include neutrophils, basophils, eosinophils, and monocytic cells are the phagocytic cells, which include macrophages and dendritic cells. Fine. So now entering into the B cell and the T cell, what is this B cell? Okay, B cells are the central an important component of an immune system which plays a major role in the humoral mediated immunity. These B lymphocytes or the B cells, they origin and mature within the bone marrow. When they leave the bone marrow, when the B cell leaves the bone marrow, it expresses, okay, on the surface of the B cell, it expresses the unique membrane receptor, a unique antigen binding receptor on its membrane of the B cell. And but that the unique receptor is called as immunoglobulin or antibody molecule or membrane bonding. To be specific, it can be called as membrane bound antibody molecule, also called as B cell receptor or antigen binding receptor. Fine. So when these B cell uh, encounters an antigen, the B cell going to get activated and differentiate into a plasma cell and to the memory B cell. So before the encountering of an antigen, before the B cell encountering the antigen, such type of cell is called as naive cell, Naive, naive cell. The naive cell is nothing but a cells, the cells which are not encounter the antigen before. So such type of cell is called the naive cell. When the naive cell first encounters the antigen that gets matches with the membrane bound antibody molecule that is on the present on the surface of the B cell, and after it binds specifically to the antigen binding site on the immunoglobulin, the B cell gets activated. The activated B cell gets differentiated into a memory B cell and the effector B cell. The effector B cell is also called as plasma cell. Memory B cell have a longer lifespan and they are going to store in the body for lifetime. And when this memory B cell get activated, when the same antigen encounters for the second time. So when so during such condition, the memory B cell gets activated and then expresses and then differentiated to a plasma cell and the memory cell again when it encounters the second when it encounters the antigen for the second time. So now, plasma cells are the cells which produce an antibody in a secreted form, and the secreted antibody will not be will live only for a few days, and they secrete an enormous amount of antibody, approximately 2000 number of molecules per second to that particular antigen encounter. The secreted antibody are the major effector molecules that play a major effector molecule of the humoral mediated immunity. Fine. So the activated B cell produces the two kind of cells. One is memory B cells and second is the plasma B cell. Plasma B cells are the cells which are going to produce an antibody in a secreted form, and those antibody is going to fight against the encountered antigen, and find the antigen will be eliminated or cleared from the body. And those plasma cells which are going to which have a very shorter lifespan and are going to live only for a few days, and so these plasma B cells do not have no do not have membrane bound antibody. So the memory B cell, which is having a longer lifespan, and this memory B cell are going to get activated when B cells are going to encounter the same antigen for the second time. So in such condition, the memory B cell gets activated and expresses the same membrane bound antibody as their parent B cell. Fine. So when you talk about the cell cycle of the B cell, how the B cell, how the male B cell gets entered into the cell cycle and gets entered into a differentiation and proliferation. So initially, the male B lymphocyte, which is having a membrane bound receptor, and they should be in a GO phase or G0 phase, where there is no activation, there is no differentiation, no proliferation. Cell remains inactive. Okay, small male B lymphocyte, or it can stay cell in the resting phase of the cell cycle or GO phase. So once it encounters antigen, once the antigen matches with the membrane bound antibody of the B cell, the B cell gets activated. The activated B cell enters into the G1 phase, okay, where it prepares the cell for DNA replication, transcription, and translation, and enters into the S phase where the DNA replication takes place, and enters into the G2 phase where the mRNA, tRNA, RNA takes place, and finally the cell is ready for the cell division, that enters into the M phase, mitotic phase. So once the cell enters into the cell division phase, it gets differentiated and then proliferates, differentiate to a plasma effector cell B cell and the memory B cell. Now the memory B cell now, enters into the resting phase of the resting phase of the cell, that enters into the GO phase of the cell cycle. And the effector plasma B cell, which are going to produce a secreted antibody in a larger number, and thereby the antigen can be evaded from the body. Fine. So this B cells, there are on the surface of the B cell, there are a lot of markers that are being expressed by every B cell, by through which we can distinguish the B cell and the T cell and the rest of the immune cells. So the molecule, some of the markers that have been expressed on the surface of the mature B cell. To say a few, B220, a form of a CD45 marker, is the marker for B cell, and then class 2 MSC molecule, then CD35 and CD21 and CD32, CD80 and CD86 and CD40. These are some of the markers, cell surface marker that have been expressed by the B cell on its surface, that helps in distinguishing the B cell from the other immune cells. Fine. So coming to the next heading, that is T lymphocytes. The T lymphocytes here, upon unlike B lymphocytes, the T lymphocytes cannot interact with an antigen on its own, on its on its own. It has to be the antigen has to be processed by an antigen presenting cell and then hand over to the T cell for the T cell activation and differentiation. How these things happen? Fine. The T lymphocytes to begin with, the T lymphocytes also origin from the bone marrow, but they get mature in the thymus, the primary lymphocyte is called thymus. They migrate, or once they arise in the bone marrow, they migrate to the thymus gland for maturation. So during the process of maturation, within the thymus region, the T lymphocytes, like B cell, the T lymphocytes also expresses the unique antigen binding receptor, unique membrane bound receptor called T cell receptor. Okay, called T cell receptor on its membrane. Okay, so here the point to be noted here is that the T lymphocyte cannot recognize the antigen alone. Okay, it cannot recognize the antigen alone. That is unlike membrane-bound antibodies on B cell. Okay, that's immunoglobulin, which can recognize the antigen alone. The B cell can recognize the antigen alone. That don't need any help. But the T lymphocytes need an assistance. The T cell receptor can recognize the antigen only when it is complexed with a cell membrane protein called MHC. The antigen has to be processed and then presented along with class 1 MSC or class 2 MSC molecule. Then only the T cell can recognize such the antigen loaded with class 1 or class 2, and then the T cell gets activated and differentiated. So the T cell needs an antigen presenting cells, and the T cell that the antigen presenting cells which are going to process the antigen by phagocytosis process, and then the whole antigen is being chewed and then degraded into small peptide antigenic peptide molecule, and these antigenic peptide molecules are going to be loaded with a class 1 MSC molecule or class 2 MSC molecule, that's nothing but a cell membrane protein. And then this loaded antigen MSC complex has been recognized by the T cell receptor of the T cell. Fine. So there are if you see there are two kind of T lymphocytes, major two population of T lymphocytes. One is T helper cell and one is T cytotoxic T cell. Okay, so those T cells which expresses a cell surface marker called CD4, CD4 plus. CD stands for cluster of differentiation. So those T cells bearing CD4 cells recognizes only the antigen that is bound with class 2 MSC molecule. Remember, the T cell bearing that is CD4 marker on its surface. Okay, those cells are expressing CD4 as a marker can recognize the antigen that is being loaded along with class 2 MSC molecule CD4 T cell. So such type of cells are called as T helper cells. There are CD4 plus act as a helper cell, and those T cells which expresses CD8 plus CD8 marker, CD8 cell surface marker on its surface, and those such type of cells can recognize an antigen that is loaded or associated with the class 1 MSC molecule. So those such type of T cell which is expressing CD8 plus marker can act as a cytotoxic cell. Can act as cytotoxic cell. Remember this? Fine. So here same here for like those T cell which have never interacted with the antigen that is loaded with the class 1 or class 2 MSC molecule are called as nail cell. When such a nail cell encounters the antigen that is being complexed with a class 1 or class 2 MSC molecule, then the T cell gets activated, gets proliferated and differentiated into a two type of cells. One is memory T cell and effector T cell. Now this effector T cell, again there are two well defined subpopulation. Those effector T cells are called as T helper cells on T cytotoxic T cell. So there is another type of cells called a T suppressor cell, which plays a major role in the cancer. So the TCD4 and CD8 cells are called as T helper cells. Those T cells which are expressing CD4 called T helper cells, and those T cells which are expressing CD8 called T cytotoxic cells. So we can actually distinguish the populations of the T cells itself from based on the markers that is present on the its surface. So those T cells expressing CD4 generally function as a T helper cell. Those cells that are expressing CD8 membrane glycoprotein marker generally function as a T cytotoxic cells. Okay, so here, so after the T helper cell, recognize, I already told you, the T helper cell recognizes and interacts with the antigen that is loaded with class 2 MSC molecule. So such type of cells, when the cell is activated and becomes the effector cell, that secretes, lot of growth factors called cytokine. So secreted, like, once the T cell gets activated, uh, after interaction of an antigen with that is loaded with the class 2 MSC molecule, the T cell expresses a larger number of cytokine, and that secreted cytokine in turn activates the B cells, the cytotoxic cell, macrophages, and other various immune cells that plays a major role in the immune response. For example, here you can see here this is an example of how the T helper cell after activation of T helper cell, how it activates the other immune cell. Example, the B cell. The B cell, which is also which acts as a antigen presenting cell, is also called as phagocytic cell. So once the B cell, bind with an antigen through the membrane bound receptors called immunoglobulin, the antigen being engulfed and then form a phagosome. The phagosome then fuses with the lysosome and resulting in a phagolysosome, and then the whole antigen is being chewed into a small antigenic peptide. So these antigenic peptides are being taken or loaded with class 2 MSC molecule. In this case, B cell, which expresses a class 2 MSC molecule, cell surface membrane glycoprotein, and the antigen being loaded with this is you can see the red color antigenic peptide that is being loaded with the brown color, which is class 2 MSC molecule. So this complex antigen MSC complex is going to be get exposed, being recognized by the T helper cell. The T helper cell recognizes the antigen that is loaded with the class 2 MSC molecule. Now the T helper cell gets activated. The activated T helper cell. Okay, remember the activated T helper cell in turn activates the various cytokines and the various growth factors. Okay, this is can see the arrow mark. It activates the expresses the various cytokine. The cytokine in turn activates B cell. That's what you can see. The activated B cell cytokine receptor. The activated B cell. Now the activated B cell produces a larger number of plasma B cell and the plasma memory cell. So that's why the immune system to see every step all are interrelated. So the activated T helper cell in turn activates the B cell. So that by the antigen being eliminated or cleared from the body. So fine. So the third type of, I told you that there is one more population of T cells called a suppressor cell, also called suppressor T cell, which expresses, okay, it expresses cell surface, unique cell surface marker. Example, CD4, CD25. Okay, so such type of cells called regulatory T cell, or you can shortly call it TREX. So these regulatory T cells, which plays a major role, suppressor cells, the immune system must discriminate between the self and the non-self. When such self and non-self discrimination fails to fails, the immune system destroys the cell and the tissues of the body, and as a result, it causes an autoimmune system. It means that the body tends to produce a lot of produces a lot of immune response against its own cell. So such type of the process, that is what the immune system, they should be able to discriminate. The immune system should be able to recognize which is self and which is non-self. Which fails to do that, the immune response start reacting against its own self cells, and that result in an autoimmune disease. These regulatory T cells, which actually suppresses the activation of the immune system, okay, against the self cells. That's what, against self cells, thereby preventing the autoimmune diseases. So the regulatory cells plays a major role, okay, in the major role in suppressing the immune system that is reacting against its own immune cells. So, and that is preventing an autoimmune diseases. Okay, so to see the summary of the T cell, which expresses a unique antigen binding receptor on the surface called T cell receptor. The T cell receptor for antigen. Okay, remember the T cell receptor of the T cell cannot recognize the antigen alone. The antigen has to be processed and then presented along with either class 1 or class 2 MSC molecule. Then only the T cell will recognize the antigen that is being complexed with class 1 or class 2. Okay, so those one after that, after recognizing the antigen and MSC complex, the T cell gets activated. The activated T cells, secretes a lot of various cytokines and growth factors, in turn, which activates the B cell for the B cell activation and proliferation into a memory B cell and plasma B cell. Okay, that way they work together. And then to see the those cells which express the CD4 as a marker, they're called as T helper cell. So the T helper cell are going to express us. You see, the T helper cell are going to recognize the antigen that is bound only with the class 2 MSC molecule. And those T cells which express the CD8 marker on its surface are called as cytotoxic. TC, cytotoxic T cell, and those cytotoxic T cell can recognize an antigen that is bound only with class 1 MSC molecule. So remember this. Okay. So I hope you are able to understand how the B cell gets activated and how the T cell gets activated, how the humoral mediated immunity plays a role, and how the cell mediated immunity plays a major role, and how they are interrelated, and how they are interrelated in activating each other, thereby bringing an immune response. Okay, thank you guys. We will in the next video we'll come up with a different topic. Thank you.